So first of all, hello everyone, and thank you for being with us today in order to join the webinar of today that is titled Regulatory Updates on Extractable and Leachables Session 1. So as the title anticipate to you, this is the first webinar of two that takes care of regulatory updates. So surely my colleague Valeria will be able to anticipate you when will be the second session. But let's start from some information as I wrote into the chat for everyone. If you have some question, any question in the middle of the presentation, feel free to use the Q&A section, okay? That is in the high part of the screen. And we will address this question at the end of the webinar. So before we deep dive into the main agenda, let's introduce our speaker of today, my colleagues Valeria Mazzoni, that is the scientific project coordinator here at Neotron. So thanks, Valeria, for being with us today. Let's now speak a little bit about the agenda of the webinar of today. So we will start with the fundamental of extractable and leachable. Then we are going to deep dive into the ICH Q3E guideline, and then updates on 661 and 662 of the USP, and some, as Valeria wrote, direction of travel waiting for session 2. So let me say an anticipation on what is next in the few months. So let's start very briefly about our company, our corporate, this Cotecna. So Cotecna is a leader in testing, inspection, and certification company. Cotecna is based on Geneva, and we have more than 6,200 employees all around the world, with more than 180 offices that takes care, as you can see from these slides, of many different fields, from inspection to certification, then to food, pharma, agriculture, metals, minerals. We have many different types of activities in order to support companies like yours. Then, if we start, let me say, giving to you some details about Neotron. Neotron is one of the business unit of Cotecna. We are based in Italy, in Modena, halfway, as I always said, from Bologna to Milan. And the lab is based on two, let me say, different plants in the same area. So we can cover 10,000 square meters of labs. We have more than 500 employees, and our technician is a very big lab on that, and we receive more than 1,000 samples per day. So we work in Italy, both in Europe, let me say, worldwide. In terms of different fields of testing, if we go to the next slide, we cover different type of activities. So we start 50 years ago with food, and then we move to baby food. We develop a supplement division, a cosmetic division, an R&D department, a sensor and consumer analysis through pharma. That will be surely, let me say, one of the topic of today. So different scope in the company to support the industry, different industry. So I think now is the time to leave the floor to Valeria, to the core of the session of today. So Valeria, the floor is yours.
So thank you, Luca. Thank you for the kind presentation, and thank you everybody to join us today. And welcome everybody. So today, as said before, I'm pleased to guide you the new release draft of the ICH Q3E and the updated USP 61.1 and 61.2 for the extractable leachable, providing a brief overview of the key points that have a significant impact on how pharmaceutical companies approach material selection, process design, analytical strategy, and risk management. So, as we saw, we have a full agenda today, and I want to start with the first introduction of the core principles of extractable leachables that will be explored, why this topic matters, what's behind the pharmaceutical product compliance, and how the regulatory landscape is managing it. And from this point, we see how the new regulation update could represent a significant evolution from the current patchwork of all guidance documents, and how it will bridge the gap between quality and safety workflows through a risk-based approach. And at the end, an overview on which will be the next steps of the regulatory updates that will be discussed in the second session of this series of webinar. So, starting with the fundamental of the extractable leachables, what are extractable leachables? Extractable leachables refer to chemical substances that may come from materials used in manufacturing, packaging, or delivery systems for pharmaceuticals and medical devices. In particular, extractables are compounds that can be released from a material when it's subjected to harsh, exaggerated conditions, such as strong solvents, elevated temperature, or extreme pH. And these conditions go beyond what the product would normally experience, but they help identify the full range of chemicals that could potentially migrate out of the materials. White leachables, in contrast, are the substances that actually migrate into the drug product or ultimately into the patient under real world conditions of storage and use. Because leachables represent the real exposure, they are the primary focus for regulatory authorities and risk assessment too, of course. And using these definitions, extractable leachables are expected to be theoretically strongly linked together. So extractable essentially serve as a worst case scenario for what could leach under real use. However, in practice, the connection between the two isn't always straightforward. And this variability often arises from specific interaction between the formulation and the packaging system, which can alter what actually migrates under normal conditions. But understanding and identifying extractable leachable is an essential key point to protect both product quality and patient safety. In fact, first, extractable leachable compounds can directly impact the quality of a drug or device because they may interact with the formulation, alter its potency, or compromise the performance of the medical device, for example. Second, and most critically, leachables can pose serious health risks to patients. Some of these compounds are toxic, carcinogenic, or can trigger allergic reaction even at very low concentrations. Third, extractable leachable can affect the shelf life and the stability of the product. So unintended chemical interaction may cause, for example, degradation over time, leading to a reduction of the efficacy or even harmful byproducts. Finally, there are real-world consequences. In fact, there have been notable product recalls due to the presence of leachables, for example. And these events not only jeopardize patient safety, but also result in significant regulatory and financial repercussions. So this is why early and thorough extractable leachable assessments are not optional. They are quite essential. So let we move to the new draft of the ICH Q3E guideline. How can we manage the importance of this topic? But I want to start with which is the panorama of the guidelines today. So regulatory bodies, including the FDA, the EMA, have established comprehensive guidelines and standards for the evaluation and control of these compounds. And here you can find some of the key standards and guidelines. For example, we have the USP 163, which outlines how to conduct a risk-based assessment of the extractable using specific simulation studies under exaggerated conditions. Then we have the USP 164 that complements that with guidance on the leachable testing. So focusing on what compounds may actually migrate under real world conditions. And for combination products, we have the USP 165 that integrates this concept with device-specific consideration, providing an harmonized approach. In addition, we have the USP 61.1 and 61.2 that we'll discuss later, focus on the characterization of the plastic materials and packaging system, respectively. And these are vital for qualifying components used in pharmaceutical containers, closures, and closure system delivery systems. Then we have the ISO Y 10993 Part 18 that focuses on the chemical characterization of the medical device materials. Moreover, on the industry side, we see valuable contribution for collaborative groups. For example, the PQRI that has published extensive guidance, particularly for orally inhaled and nasal drug products where patient exposure risk is quite high. And similarly, the Bioforum Operation Group, the PBOG, provides the best practices for evaluating single-use system in biologics. The risk-based models are often referenced in regulatory submission. But despite the essential role that extractable leachable evaluations play throughout pharmaceutical development and product lifecycle management, the regulatory environment, as you can see, has long been disjointed. And until recently, there was no single unified global standard that clearly defined how extractable leachables should be identified, toxicologically qualified, and controlled using a risk-based approach. So this lack of harmonization has posed major difficulties for global pharmaceutical companies, resulting in inconsistent practices, differing interpretations of expectation, and considerably regulatory uncertainty. So a significant step forward came on the 1st of August of 2025, when the International Council for Harmonization introduced the draft of the Q3E guideline on extractable and leachables. And this marks a significant milestone in the global harmonization of control, specifically addressing the extractable leachables in drug products and drug device combination products. Building on existing ICH guidelines, the Q3E provides a comprehensive framework applicable to pharmaceuticals, drug device combination products, and advanced therapies, biologics, cell and gene therapies, combination products, and single-use manufacturing system have created, let me say, extractable leachable challenges that existing guidance documents were never designed to address. So today we have an important opportunity to elevate global standards for the extractable leachables. By establishing an harmonized framework, we can bring clarity, consistency, and scientific rigor to an area that is fundamental to patient safety as the main point. And our goal is to align current expectations so that the scope is clearly defined and the focus remains on the most critical quality and safety aspects. a unified framework will enable threshold for reporting, identify and qualifying extractable leachable to be guided, considering the route of administration, the therapeutic indication, and the actual patient exposure. It will allow us to address existing gaps and complement current ICH guidelines by providing much needed direction where guidance is still lacking. Equally important, we must strengthen the foundation for safety assessment. This means defining relevant safety limits based on the exposure scenarios and clarifying how prior knowledge can be used to determine when additional studies are truly necessary. Ultimately, by improving consistency between regulators and industry, we not only streamline development, but also reinforce our shared commitment to patient protection. So together we can build a coherent, globally accepted approach that serves public health and support innovation too. The most transformative aspect of this guideline lies in its integration of comprehensive risk management principles that derive from the ICH Q9 throughout the entire extractable eligible assessment process. And this represents a fundamental departure from the prospective one-size-fits-all approaches that have characterized previous guidance documents. And in this field, the risk management framework encompasses four critical stages: the hazard identification, the risk assessment, the risk control, and the life cycle management. And in particular, risk assessment is composed by three different steps. So the hazard identification, the risk analysis, and integrated risk evaluation. So let's see them in the details, in the deepness. The effective control of extractable eligible begins with a robust hazard identification, and this requires a deep understanding of each component's potential to interact with the formulation, as well as the impact of any pretreatments or processing steps that may alter material behavior. And by anticipating these sources of risk early, we create a solid foundation for a science-driven evaluation. Next, we move to the risk analysis, where real-world factors come into play. So manufacturing conditions, storage environments, and the intrinsic leaching propensity of the formulation all influence the likelihood and the level of the patient exposure. So assessing these elements together allows us to distinguish theoretical risk from those that are truly relevant. And finally, integrated risk evaluation brings all dimensions together. So linking the potential hazard severity with the acceptable risk thresholds. And this step enables us to weigh the scientific evidence, determine whether additional mitigation is needed, and justify decision transparently and consistently. And through this structured approach, we ensure that the product ultimately achieves the fundamental goals of the pharmaceutical development. So the uncompromised quality, the assured safety, and then consistent efficacy for every patient. But we have to manage different points for the risk evaluation, and the multifactorial risk matrix introduced in the ICH Q3E is firmly rooted in the principle of the quality risk management. It recognizes that the evaluation of extractable leachables must be systematic, science-driven, and proportionate to the risk. So, in this framework, pharmaceutical quality critical dimension form the foundation of the risk assessment. And we begin by examining the potential for interaction between manufacturing equipment or container closure system and the drug product, of course. And this includes understanding the physicochemical properties of each component and any pretreatments that may influence their behavior. Quality risk management therefore requires consideration of all process and product variables that can affect the leaching. manufacturing and storage conditions such as the surface area to volume ratio, the temperature, the duration of the contact play key roles. And equally important is the formulation intrinsic leaching propensity. So the reactivity of the API, the pH, the organics, co-solvents, surfactants, and chelatic agents all determine together how readily compounds may migrate into the drug products. And once the quality dimension are understood, the safety assessment overlays these findings with patient-centric factors. So here, the potential safety impact of the leachable is evaluated in the context of route of administration, specific patient population, maximal dosing, dosing frequency, and lifetime treatment duration. So these exposure-related variables define the clinical relevance of any leachable detected. And by integrating this quality and safety dimension into a cohesive multifactorial risk matrix, the ICH Q3E provides a transparent and scientifically balanced tool, ensuring that every decision is consistent with quality risk management principles, supporting the protection of patients through reliable, justified, and evidence-based evaluations. And starting from this consideration, we can, let me say, visualize the risk assessment process as a tower that is solid, structured, and built step by step from its most fundamental pieces. And at the base of this tower, three main blocks lie. So the manufacturing conditions, so the temperature, the contact duration, the surface area to volume ratio, the storage environment, and so on. And these define the physical reality of how materials interact with the product and set the groundwork for understanding leaching behavior. Then the second point is the composition of material themselves. So their physical chemical properties, any pretreatments, the stability of each component that shape the potential for extractable and leachables. And this layer relies heavily on the quality and completeness of available data. So previous studies, material characterization, and historical knowledge can either strengthen or weaken this tower. And then the next layer, consider how the product is intended to be used. So formulation characteristics, the route of administration, the duration of the use, all together influence the likelihood and the relevance of the patient exposure. And finally, at the very top of the tower, we place the patient. So the extent and the nature of the patient contact, the vulnerability of the intended population, dosing frequency, and the potential lifetime exposure. And this final piece determines the true significance of all risk identified below. So when these layers are highlighted in tower, becomes a current science-based risk assessment. And it is this structure approach that ensures our decision that must be justified, defensible, and proactive, protective also to the patients. And as anticipated, the Q3 guideline emphasizes that the control of extractables and leachable must follow a true life cycle management approach. As products evolve, so in the same way, thus our understanding of the factors that may influence patient exposure and benefit risk. So whenever new information emerges, a regulatory impact assessment becomes essential to ensure continued patient protection. So several types of changes can trigger this reassessment. So here, a list of these changes that can deeply affect the risk assessment and the risk management in general. So any modification to the drug product formulation, whether through altered excipients levels, the pH adjustment, or the introduction of any co-solvents, can meaningfully affect the leaching profile. Likewise, updates to the packaging components or the entire container closure system may introduce new materials, new suppliers, or new configurations that need evaluation. And importantly, the ICH Q3E also highlights changes related to manufacturing process. So any adjustment to the manufacturing process or to any components and system that can come into direct contact with the drug product or drug substance can alter material compatibility and the chemical migration. So these factors may increase or decrease or shift patient exposure in ways that must be scientifically assessed. And through this risk-based lifecycle framework, the ICH Q3 ensures that evolving evidence is continuously integrated, that regulatory expectations are met, and that the benefit-risk profile of the product remains controlled and transparent. And above all, it reinforces our commitment to safeguarding patients throughout the entire life of the product. However, risk assessment related to risk identification and risk mitigation must be distinguished between manufacturing components and systems and primary packaging and delivery device component system. So in the first case, we can see that the attention is focused on the extractable evaluation. That is the core of the entire assessment. And starting from this main point, three different scenarios could be depicted. As you can see, we have, for example, for solid or a drug product and manufacturing, no extractable leachables is needed. We have the optionable extractable leachable evaluation for liquid oral drug product using polymeric manufacturing equipment. No leachables if we have no manufacturing component system with extractable above the specific toxicological threshold fixed. And all manufacturing equipment extractables detected, identified, and quantified in the quantitative extractable study above the applicable toxicological threshold that are below their applicable safety thresholds. While for the, in the second case, for the packaging and delivery device, the risk assessment takes another aspect, one that is much more complex. In fact, in this workflow, the necessity to work in a combination field between extractable and leachables is fundamental, recalling the main risk management matrix with the two main dimensions. So pharmaceutical quality, so with interaction potential between the components, the formulation, the material properties, and so on. And the patient safety, so the route of administration, the patient population dosing regimen, the treatment duration, and so on. So moreover, a correlation between the extractable and the actual leachables should be established whenever possible. For example, unexpectedly high leachable levels compared to extraction prediction may indicate an incomplete extraction study design. This means that the design of the study plan of the extractable and leachable evaluation must be defined in the correct way. And in this case, we have three scenarios that could be depicted. So as for the manufacturing components, we have no extractable leachables for delivery device components with very short or transient contact with oral drug products. We have the optionable extractable leachable study for containing closure system components for oral drug products that are compliant with regional food contact regulations, including composition, fabrication, specification, testing result, and so on. And extractable leachable are needed for frozen, no lyophilized drug products stored in a well-characterized packaging system. So we have different scenarios that we have to deal with. But the ICH Q3E introduces a clear and structured workflow for the safety assessment of leachables, built on a tied approach that aligns the depth of the evaluation with the inherent hazard of each compound. So the process begins with the classification of the leachables. So the class 1 leachables are those with the highest toxicological concern, often associated with the structural alerts or limited safe thresholds. And these require immediate attention, so full identification, and the application of TTC or PDE or other toxicological limits to ensure patient protection. While for class 2 leachables, represent a moderate level of concern. Here, the identification is still required, but the assessment relies on existing toxicological data, literature, and read across, or in silico tools. And their acceptability depends on heavily on exposure factors such as the route of administration, the dosage frequency, and the duration of the treatment. And finally, class 3 leachables are generally viewed as low concern compounds with well-established safety profiles. And at typical low exposure levels, identification and prior knowledge are often sufficient for the qualification. However, when class 3 leachables exceed 1 milligram per day, the ICH Q3E requires a deeper compound-specific safety evaluation. And at these higher levels, prior knowledge alone is no longer adequate. So toxicological data, PDE derivations, and detailed consideration of route of administration and lifetime exposure must guide the acceptability of these compounds. And by applying this stepwise workflow, so class with the strictest scrutiny, the class 2 with a contextual evaluation, and class 3 with minimal additional requirements, ICH Q3E ensures the science-based, transparent, and proportionate approach to safety assessment. So this methodology supports consistent, defensible decisions and reinforces patient safety across the product lifecycle. So, in summary, which are the main points that this guideline reinforce? For sure, the global harmonization achieved that addresses one of the most significant challenges facing in the pharmaceutical industry by providing consistent requirements and expectation across all major regulatory jurisdictions. The transformation initiated by this guideline extend beyond technical requirements to encompass fundamental changes in how pharmaceutical companies approach the material selection, the process design, the analytical strategy, and risk management. And the focus must be also on building the scientific expertise, the analytical capabilities, and the quality system that is necessary to realize the guidelines potential for enhancing patient safety. So, moving on our agenda, we saw that the ICH Q3 tell us what risk we must evaluate, but how to know if the packaging materials are qualified. And in this case, the USP 61.1 and the 661.2 tell us how to test the materials involved and provide the material qualification test needed to demonstrate this compliance. So, moving on, we can go deep into this topic. While the ICH Q3 establishes the global expectation for how extractables and leachables should be assessed through a science and risk-based approach, manufacturers must also ensure compliance with region-specific compendial requirements. And these expectations are defined in the USP chapter 661.1 and 661.2, which together replace the older and far less specific USP 1. And as of December the 1st of 2025, the USP 61 has officially become obsolete, and compliance with the new chapter is now mandatory. So these shifts represent a significant tightening of regulatory expectation, as the USP 661.1 focuses on the qualification of the plastic material of constructions, while the 661.2 addresses the suitability of the final packaging system. And regulators now expect manufacturers to generate data aligned with these new chapters to demonstrate that packaging components are appropriate for their intended use and compatible with the drug product. In practice, this means that the risk-based assessments guided by the ICH Q3E must now be supported by standardized material testing defined by these two USPs chapters, and together these requirements ensure not only scientific rigor but also consistent and verifiable proof of packaging suitability throughout the product life cycle. Starting in 2013, a large push has done to update package-related chapters in the USP during these years, as you can see. And since the first step, every single chapter has been impacted or updated in some way. And as you can see, they span all different types of materials. As you can see, elastomers, plastic, glass, and so on. And there are general test chapters that introduces all details of analytical insight and information chapters that helps providing the context of the tests that have to be performed, how you consider those, and interpret the relative results. But moving forward, what changes, which are the changes between the old USP 61 and newest 61.1 and 61.2? So here you can see in this table in details the main points of difference between all these chapters. So we can see that the scope is different because we move from a broad test for plastic packaging of the 61 to a material raising specific characterization for the 61.1 and a full system suitability for the entire packaging system as a whole for the 61.2. We have a different focus. In fact, we move from a general fitness to a specific identification of physiochemical bioreactivity of the resin and the first, and the chemical suitability for the whole packaging system. We have a different risk-based approach. So we move from an implicit approach to a risk assessment, a risk-based assessment that is linked to the ICH Q3E mindset, let me say. And of course, we have a different status. So a retired USP and an official mandatory USP 61.1 and 61.2. But going into the deep of each chapter, USP 661.1 gives us a clear pathway to ensure plastic materials used in the pharmaceutical packaging that has to be fit for the purpose. So it starts with polymer characterization standards. So we verify who the materials is and how it behaves by conforming identity, understanding the key physical chemical properties, and screening for any biological reactivity. With this baseline, we know exactly what we are putting in contact with the drug. So next comes the essential testing. Here we translate the standards into an evidence. So material identity verification, the compositional analysis, and the targeted physiochemical assessments, such as heavy metals, residuals, and other critical quality attributes. And these tests don't just generate data, they establish specification and provide a reliable benchmark for consistency over the product life cycle. Finally, biocompatibility connects material science to patient safety. And by aligning the test strategy with dosage form and intended use, we evaluate whether the plastic is suitable in real world conditions, supporting the risk assessment, the change control, and ongoing compliance. So together, polymer standards, essential testing, and biocompatibility form, let me say, like a coherent science-based framework that safeguards the packaging suitability and protects the quality, the safety, and the performance of the drug, of course. While for the USP 661.2, it defines the expectation for evaluating the suitability of complete plastic packaging system used for the pharmaceutical products. And its scope is broad, covering containers, closure system, droppers, tips, blister, laminates, and so on. So all material that comes into direct contact with the drug product. And by setting clear requirements, the chapter ensures that every part of the packaging system is fit for purpose throughout its intended use. So to demonstrate suitability, these chapters require a packaging system to meet rigorous assessment criteria, and these include the physiochemical properties to confirm material consistency, the biological reactivity test to evaluate the interaction with the living systems, and the chemical safety assessment to assure that extractable and leachables remain within acceptable limits. And the goal is to ensure that the packaging system maintains product quality and does not compromise the patient safety. So, most importantly, USP 661.2 adopts a risk-based approach, and the depth and the extent of testing are aligned with the intended use of the packaging in the dosage form and the potential for patient exposure. And as you can see, this approach is fully aligned with the principle of the ICH Q3E, ensuring the extractable leachable assessment are scientifically justified and proportionate to risk. But implementing these chapters requires a structured workflow that ensures packaging systems are fully understood, qualified, and controlled throughout their life cycle. And the process begins with collecting all resin data available, gathering all information on material composition, additives, and prior testing. And this forms the foundation for understanding how the polymer behaves and whether it meets the identity and characterization standard of the 661.1. Next, we map the entire packaging system. So this means documenting every component that contacts the drug product, from containers and closures to liners, tapes, and secondary interfaces. Clear mapping helps determine which elements require testing and the level of scrutiny needed. With the system defined, we move into the physical, chemical, and biological reactivity testing. And this assessment verifies material integrity, evaluates extractable metals and residues, and ensure the plastic does not exhibit adverse reactivity. And this is where the requirements of both chapters transition into the system level expectations. Then we move to the chemical suitability. That's is assessed by integrating extractable leachables data, dosage form consideration, and risk-based principles aligned with the ICH Q3E and USP 163 and USP 164. And the goal is to confirm that the packaging system is safe and comprehensive with the drug product under the real world condition. And finally, all findings are consolidated into a comprehensive dossier, which becomes, let me say, like the basis for life cycle management. And this ensures that any future material or process changes can be assessed transparently and consistently. So together, these steps form a coherent and defensible workflow for demonstrating the full compliance of the materials and the packaging. So in conclusion, when we look at extractable and leachables control across global and compendial expectation, it becomes clear that true compliance is achieved only when we integrate the strengths of each guideline into a single risk-based framework. So the ICH Q3E provides foundation by defining how to evaluate the risk. So it guides us through the hazard identification, the exposure assessment, and the science-based qualification of leachables. And this establishes the core principles for determining what matters most for the patient safety. While the USP 661.1 then complements this foundation by requiring details for the racing characterization, and it provides the material level understanding that feeds directly into the ICH Q3E. And these data enhance predictability, can reduce also the uncertainty and help determine the likelihood that extractables may migrate into the drug products. And finally, the USP 61.2 brings a system level evaluation, confirming that the complete packaging configuration is suitable for its intended pharmaceutical use. And this chapter transforms the material data of the 661.1 into a real-world evidence of the packaging performance aligned with the dosage form, the route of administration, and the product life cycle. So together, the ICH Q3E, the USP 61.1, and the USP 61.2 create an integrated compliance framework for which risk principles define what to assess. So the racing characterization provides the foundational data, and the system evaluation confirms suitability. So this combined approach ensures a defensible, harmonized, and patient-focused risk-benefit assessment. But what's next? In this year will be, let me say, full of news and update information. The first is the progression of the ICH Q3 toward the finalization. So we will have the global harmonization for the extractable leachable expectation with alignment with other all these guidelines, ICH guidelines, and with increased emphasis on the system-wide extractable leachable evaluation and life cycle control. Then we will have the expansion of the USP 164 chapters. As you can see, we will have four different chapters that we discuss and evaluate leachables in different pharmaceutical products. So we have the leachables in orally inhaled and nasal drug product with a revision of the 164.1. The leachables and in parental products, new for a new chapters, as for the topical ophthalmic products and topical and transdermal products. Then we will have an implementation of the USP 65 in the 1st of May of this year. The status will be informational, and considering the current ICH activity, and USP is working to review all chapters related to extractable leachables to ensure the complete alignment with the ICH Q3 guideline. And we will see an increasing scrutiny for the single-use materials, the device-associated leachables, and the upstream process contributions. And finally, we don't have to forget also the medical devices. And we, in this year, will have the MDR bottleneck between 2026 and 2027. We will see a comprehensive chemical characterization aligned with the updated ISO 10993 chapter 1. We will see that how hopefully a strong justification connecting extractable to biocompatibility claims, and a detailed toxicological risk assessment for all leachable. So for all of this news, see you on May for our session 2 of this series of webinar. So now we move to the Q&A.
Thank you very much. Thank you very much, Valeria. It was a very comprehensive session, and also I learned new things from you today. So thank you very much. As you anticipated, it's now the moment of the question and answer. So we'll open the floor. You can type any type of question in the question and answer box, but we will start by trying to reply at your question that that arrives during the registration period. So we will start reading the first one that we received during that. So the first one is, what are the most common extractable and leachable related deficiency regulators see in submission, and how can companies proactively avoid them? Please, Valeria.
Thank you, Luca. Okay, this is quite a huge question. It's a wide range question. Let's walk through the most common extractable leachable deficiency regulators that continue to highlight and what we can do to proactively prevent them. First, one of the biggest issue is incomplete extractable studies. So regulators frequently see studies as leave out relevant contact materials, especially from manufacturing equipment, or that don't apply adequate worst case extraction conditions. And this has been clearly noted in FDA assessment discussion, where missing polymeric surfaces and insufficient extraction design complicate the review process. Another recurring deficiency is missing or insufficient leachables data, leading to regulatory questions or post-marketing commitments. We also see problems with risk assessment that only evaluates individual components rather than the entire system. And regulators emphasize that reviewing components isolating is not enough. So leachables, as you know, are cumulative, and contribution can add up across the full manufacturing and packaging workflow. Another area of concern relates to the toxicological justification. Regulators expect a clear rationale for safety thresholds and appropriate toxicological assessment for each detected compounds and leachables, of course. Weak or incomplete evaluation are a common reason for deficiencies. And then there are the analytical method limitations. For example, methods that lack the required sensitivity, or that don't fully identify unknown peaks, or that haven't been shown to be fit for the purpose. And these problems directly undermine the reliability of the extractable leachable data packaging. And surprisingly, a common issue is a poor organization of the extractable leachable information. In fact, when data are scattered across multiple modules, it slows down reviewers and increases the likelihood of follow-up questions. And the FDA, for example, now specifically recommends the use of a extractable leachable summary to make submission clearer and more efficient. So how can we avoid these problems? for sure, we have to design comprehensive worst-case extractable studies. We have to adopt a fully life cycle system-wide risk assessment. We have to invest in a strong toxicological evaluation and provide a well-structured extractable leachable summary to streamline the regulatory review and reduce the back and forth. So overall, these proactive steps, it can help ensure that extractable leachable programs are compliant, scientifically robust, and aligned with the evolving expectation that we see are quite important.
Thank you, Valeria. A very complete answer to this complex question. Thank you. So we can move to the second one that we receive is how does ICH Q3E change the way companies approach extractable and leachable risk assessment?
Yeah, as we saw, the ICH Q3E reshapes how companies approach extractable leachable by introducing an holistic, risk-based, and system-wide framework for assessing materials interaction. And the guideline requires companies to evaluate all contact materials cumulatively rather than performing isolated, as said before. So we don't have to consider the component level assessment. So we have to address the cumulative components. And in order to have formal risk management principles, managing the testing scope, extraction condition, and leachable study decision that must be justified and fully documented. So the guideline strengthens the link between the material characterization, process understanding, and the patient exposure, requiring companies to ensure extractables data meaningfully predict real-world leachables under the manufacturing and storage conditions.
Thank you, Valeria. So we go to the third one. So why are the 2026 updates to USP 661, 662 important?
These updates are critical because they replace the older, more general USP 661 with, let me say, a rigorous risk-based framework that strengthens control over plastic materials used in the pharmaceutical packaging. And these two chapters mark a significant shift in expectation for both packaging suppliers and drug manufacturers. The 61.1 requires comprehensive characterization of the plastic materials. And on the other side, the USP 61.2 focuses on the chemical suitability of the complete packaging system. And these integrate extractables and leachables expectation in line with the other USP chapters, so the informative 163 and 164 framework, ensuring that the packaging performance and its chemical interaction with the drug is evaluated in an holistic way, not just at the raw material stage. So these updates reflect the industry-wide move towards a stronger patient safety, improving the product integrity and the globally harmonized standards, addressing the long recognized gaps in the previous chapters. So adopting these requirements, companies gain a more predictable and robust foundation for packaging qualification and reducing a lot the compliance risk.
Thanks, Valeria. So we move to another one. So why is alignment between regulatory expectations and extractable and leachable practices so critical in 2026?
The alignment of the extractable and leachable practices with regulatory expectation is more critical than ever because the global framework for extractable and leachables has evolved significantly with the introduction of the ICH Q3E, which establishes an unified, holistic, a risk-based approach that assess the extractable leachable impurities across all dosage forms and contact materials. So these new guidelines fills long-standing gaps and aims to reduce uncertainty for both industry and regulators, as previously inconsistent expectation often led to a variability of the interpretation and the approval delays. So at the same time, regulators continue to highlight the common deficiencies, such as the incomplete extractable studies, the missing leachable data, and so on, which contribute directly to the review of the challenges and regulatory questions. So staying aligned ensures it's quite important because it ensures regulatory confidence, smoother approvals, and stronger patient safety insurance, reducing all the rework, and all of which are essential as the regulatory environment becomes more harmonized and demanding during this year.
Thank you very much, Valeria. So just
Thank you.
looking at the time, we are a few minutes in delay, okay, than was the previous set time for today. So we are going to reply to all the other questions by the Q&A document that we receive in the next day. So just to we are at the conclusion of the program of today. So as I was anticipating to you at the beginning of the session of today, we will send to you an email in the next coming days containing the recording of today's sessions and also the Q&A document, the presentation, and a survey of the webinar of today. Surely there will be also a link on the subscription newsletter of Cotecna and Neotron. We organize these webinars frequently, so you are surely welcome to participate all the others. So I was your moderator of today. I'm Luca Vogesi, and I'm in charge of marketing and business development here in Neotron. I hope that you find this webinar beneficial, and surely a big thanks to you, Valeria, for the knowledge that you spread today to all of us. I hope that you find you all beneficial today. And what else to say? Have a great day, everyone, and see you in the next webinar in session 2 of Extractable and Leachable. Bye, and have a nice day.